Hi, Blazers. Here with the last part of this section on photosynthesis. So this is the first half of the unit on what we call energetics. So we'll finish up photosynthesis today with the dark reactions or the light independent reactions. And then next week, I'll go to the second part of energetics, which is cellular respiration. All right, so the light independent reactions, take place not in the thylakoids, the way the light dependent reactions take place, but in the stroma or the liquid portion of the chloroplast. So they're going to be in the part of the photosynthetic reaction that we call the Calvin cycle. It was discovered by a person whose last name was Calvin, so he named it after himself. The big thing about the Calvin cycle is that here's where our carbohydrates are going to be. 6 carbon sugars are things like that we will produce during this cycle are going to be made. Right? So we said earlier that the light-dependent reactions take place here in the thylakoids. And what we're going to see is the light-independent reactions are going to take place out here in the stroma. Right? And this is where we're going to find all of our enzymes that we're going to need in order to have this reaction take place. So remember, we're calling this a Calvin cycle. And the reason we call it a cycle is because it is circular. And what we're going to see is we're going to use energy that we're going to get from the synthesis of ATP in order to go through really the 3 stages or steps that you're going to see in this particular cycle here. And those 3 stages are fixation, reduction, and then the regeneration here that we're going to find. So we start with a very key molecule that is going to get this whole thing off and running, and we call it ribulose, and you'll see it abbreviated as RUBP if you look up here. And it's going to be very key because it's going to take that carbon dioxide from the light reaction, and it's going to use it to keep that cycle churning or regenerating. So the first enzyme that we're going to encounter is what we call Rubisco, and it is a 5 carbon. You see here 1, 2, 3, 4, 5 carbon enzyme, and here with Rubisco we are going to carboxylate that RUBP that we talked about earlier. All right, we split using water, we split this up, and what we end up with are 2, really 3 carbon molecules. And so we end up with our 6 carbons, 1, 2, 3, 4, 5, 6. So we get those 2 right here. Here's 1 3 carbon molecule. Here's the second 3 carbon molecule. Okay. This 3 carbon molecule is our glycerate over here. So that's what we're going to be looking at, our phosphoglycerate molecule. And this 3 indicates that there are 3 carbons. So we take that glycerate phosphate, or GP we're going to call it, and reduce it using this ATP. So here's our GP, here's 1, here's 2, here's our ATP, coming in. And whenever you see ATP, you know that there is energy that is going to be used up in this particular reaction. So we take those two, we break down that ATP, we lose one of the phosphates, and we release our ADP, adenosine diphosphate, and we get closer really to the type of carbohydrate that we need. Remember, there's a very specific ratio of 1 to 2 with carbons to hydrogens in our carbohydrates. All right, so taking a look at the second part here, the regeneration piece. We had in stage 1 that fixation that occurred, then we had the reduction, and now we're into the third phase, which is the regeneration piece. So this whole piece is what we consider to be the dark reaction or the light independent reaction, all done inside of the Calvin cycle. Okay? So our carbon dioxide, 3 molecules of carbon dioxide come in from the light side, combines with our Rubisco and gets us rolling in through the fixation of our carbon, and then the regeneration after the reduction of our carbon. Now those remaining TP molecules, those are going to be converted into our sugars or starches. That's where the carbohydrates come in. So we need really 6 turns of this cycle in order to produce what we call a hexose molecule, which means that it has 6 carbons in it. And a good example of that is that monosaccharide that we're going to produce, or glucose. So the plant gets its energy source here in the form of glucose, or this the food that's going to keep the plant going. Just another little look here, fixation, reduction, regeneration. Those are the 3 pieces of the Calvin cycle. And then there's a nice summary here for you. So please take a look at that when you can. And if you have, don't get bogged down by the names of all of these, because those are not going to be specifics like that are not going to be something that IB will be asking you on the final exam. So sometimes people try and learn all these different pieces and what they mean, and really you don't need to do that. If it's something that information you would need on that, you would look that up. So take a look at that when you can. And that's our 2 reactions. I have a nice little video clip for you that summarizes all of photosynthesis, the light reactions here in the thylakoid membrane, and the light independent or dark reactions that are going to take place in the Calvin cycle out here in the stroma or liquid. Right? One feeds the other. And in order to keep the plant going and keep photosynthesis going, we need to have both pieces, the light and the dark reactions, working together. If you have any questions, or here at the end here, just some basic information to know. If you do these, you'll be in good shape. All right, if you have any questions, just let me know. Thanks, Blazers.